Greetings for the day. I'm Soundarya from Shastra University, here to present our work titled Coarse-Grained Modeling of Human Kinesin GIF11 with its inhibitors at different allosteric sites and implications in its activity. Let me start with the introduction on GIF11, a member of kinesin 5 family. It is a homotetramer that travels towards the plus end of the microtubule. It is an N-type motor which has the motor domain towards the N-terminal region of the protein. GIF11's movement is fueled by ATP. During cell division, it plays an important role in the formation of the spindle fibers and the separation of the sister chromatids. It also plays a role in cargo transport in neuronal cells. GIF11's vital role in maintaining the microtubule dynamics during cell division makes them an attractive chemotherapeutic target. GIF11 is found to be overexpressed in certain type of cancer like breast cancer, hepatic cancer, prostate cancer, etc. During mitosis, the GIF11 depleted cells form monastral spindles, which ultimately leads to apoptosis by activating spindle checkpoint. Many of GIF11 inhibitors are at various phases of clinical trials, with neutropenia being the most common dose-limiting toxicity. They show no neurotoxicity and are shown to be highly effective by demonstrating total tumor regression in mouse xenografts. The motor domain of GIF11 has two binding sites. The nucleotide binding site made up of switch 1 cluster, switch 2 cluster and P loop, and the microtubule binding site made up of loops L11, L12, helices 4 and 5. These sites do not show any stringent specificity for inhibitor binding, but they inhibit or slows down the ADP release. The allosteric site 1 of GIF11 is made up of loop L5, alpha 3 and alpha 2, and it is the most frequently targeted. So this study is carefully designed in a way that the structure from each category is considered to study the critical structural dynamics that happen at the important regions upon inhibitor binding at different allosteric sites, that is allosteric site 1 and 2. Two structures showing the initial and final structure of GIF11's motor domain were taken as controls, and intermediate structure is also taken. A structure with inhibitor bound at allosteric site 1, a structure with inhibitor bound at allosteric site 2, a structure where the inhibitor is bound simultaneously to both allosteric site 1 and 2 were taken for analysis. Moving on to the results. To identify the regions within the protein that are positively and negatively correlated in their dynamics, genome cross-correlation maps are plotted. The red-colored regions indicate the positively correlated areas, and the blue-colored regions include the negatively correlated regions. In the first mode, we can see that the model C is more mobile with much fragmented domains. The three inhibitor-bound models show different dynamics, and upon careful observation, we could say that the first dynamic domain of model F is much similar to the first dynamic domain of model D. And the second dynamic domain of model F is much more similar to the second dynamic domain of model E. This shows that the model F has the mix of dynamics of both model D and model E. The ANM of the first three slowest modes were visualized together, and it was observed that the non-functional loop L10 was found to be more dynamic and flexible in comparison to the rest of the structures in all the 6 states. The loop L10 is found in closer proximity to the neck linker, and its dynamics can influence the neck linker's conformation. In the inhibitor-bound systems, we could also see that there is a loss of flexibility or gain of rigidity in all the functional regions upon inhibitor binding. The global cross-correlation heat maps constructed using the all non-zero modes shows cross-correlated residue fluctuations. Although the overall cross-correlation heat maps look identical, there are slight differences in between them, which are highlighted in yellow circles. Besides the global motions, the first 5 fastest modes were analyzed individually for the 6 GIF11 models. The residues with high fluctuations at the fast modes are called as hotspot residues, and they are said to play a vital role in protein folding, stabilization of key interactions, functionality of the protein, etc. These are the residues that are found in the functional sites of the protein, which could be important in binding of the motor domain to the microtubule or binding of the motor domain residues to the incoming inhibitors. PSN was done to investigate the structural communication in proteins upon inhibitor binding. The results showed that the model C has a network of structural communication with many residues, and all other systems are much more rigid, with the network being disrupted into shorter paths upon inhibitor binding. Taken together, our observations suggest that there are different mechanisms at play in each inhibitor bound system considered. The mechanism of binding at allosteric site 1 and 2 are unique, and the binding at both allosteric sites has a mix of both structural interactions, leading to a totally different mechanism of binding. Thank you.